4.5% of all cancers worldwide are attributable to human papillomaviruses, accounting for 70% of cervical cancer, 30% of oropharyngeal cancer, and almost 90% of anal cancer. HPV infections are typically resolved. However, if not cleared, HPV-infected tissue can develop epithelial hyperplasia and progress to precancerous and cancerous lesions. This hyperplasia leads to an influx of immune cells, notably of antigen-presenting cells, which are key in priming the cytotoxic T-cell response. In infected epithelium, however, APCs develop a regulatory-like phenotype, preventing the body from clearing the virus. To study the immune tolerance associated with HPV infections, we utilized a transgenic K14E7 mouse expressing HPV oncoproteins that cause hyperproliferative epithelium in ear skin. Characterizing the myeloid compartment in K14E7 skin remains an essential step in further understanding the mechanisms that lead to tumor immune tolerance and evasion. In order to study the immune landscape of K14E7 skin, we developed a 19-color flow cytometry panel consisting of 15 phenotypic markers and 4 functional markers, denoted in purple, to identify 7 myeloid populations and measure expression of functional surface features between K14E7 and wild-type skin. Samples were pre-gated on time, forward and side scatter, 2 singlet discrimination gates, a CD45 positive live dead negative gate, and CD3 exclusion to emit T cells. Here we discuss the gating strategy used to identify each of the populations and shows the comparison between wild-type and K14E7 for each population. Note the global influx of immune cells in the K14E7 sample. Following pre-gating, we first identified neutrophils, then plasmacytoid dendritic cells, and monocytes. Following this, we identified monocyte-derived dendritic cells, classical dendritic cells seen at the bottom of this plot, and finally, Langerhans cells. Of the 4 functional features measured in this panel, only co-stimulatory marker CD86 in Langerhans cells showed a substantial upregulation in K14E7, verified by an FMO for both wild-type and K14E7 samples. In conclusion, the K14E7 epithelium displayed an influx of myeloid cells, including plasmacytoid dendritic cells, neutrophils, monocytes, monocyte-derived dendritic cells, classical dendritic cells, and Langerhans cells. The K14E7 Langerhans cell phenotype differed from the wild type, displaying lower levels of CD24, CD11b, and EpCAM. Langerhans cells also demonstrated an increased expression of co-stimulatory marker CD86 in the K14E7 epithelium. K14E7 epithelium provides a useful model of precancerous epithelium, and future work aims to further investigate the myeloid immunophenotype in this model to better understand immune tolerance.